The overall goal of this procedure is to study the glial heterogeneity influence on DRG axon growth and cell adhesion. This is accomplished by first culturing the glial cells from neonatal rat cortex. The second step of the procedure is to culture and purify the rat embryo DRG neurons. The third step of the procedure is to add the purified DRG neurons onto the glial cells. The final step of the procedure is to observe the axon growth and cell adhesion and carry out cell staining and quantification analysis. Ultimately, results can be obtained that show DRG neuron growth is influenced by heterogeneous glial cells through time-lapse recording and immunofluorescence microscopy.
Hi, I'm Han-peng from Bologna, the Laboratory of Neuroimaging at the Department of Neurology in UCLA. Today, we will show you a procedure to study the glial cell heterogeneity's influence on axon growth in a new coculture method. In our laboratory, we use this new method to study the interaction between neurons and the glial cells. So, let's get started.
In order to establish a coculture of glial and dorsal root ganglion neurons, or DRGs, we will begin by culturing glial cells. Under a tissue culture hood, line the bottoms of pre-sterilized 24-well culture plates with sterilized round glass cover slips. Coat the cover slips and the wells of a 6-well culture plate